So greeting everyone, welcome to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center. My name is Martine Roussel, a member of the Department of Tumor Cell Biology at St. Jude Children's Research Hospital. So today it is my distinct pleasure and honor to welcome our speaker, Dr. Stefan Pfister. Dr. Pfister is well known to the St. Jude pediatric brain tumor community. He and I met several years ago during one of our annual medulloblastoma in the mountains meetings held in the most beautiful skiing places in the world. And I have since collaborating on several projects with Dr. Pfister. Dr. Pfister has kindly offered to stay on for some additional Q&A after the top of the hour, if anyone would like to stay for further discussion. So now I'm pleased to introduce Dr. Pfister. He obtained his MD degree from the Universities of Tübingen and his clinical education at Mannheim and Heidelberg universities. He did 2 postdoctoral fellowships, one at the Dana-Farber in the Division of Immunology with Dr. Christopher Rudd in Boston, and one at the German Cancer Center in the Division of Molecular Genetics with Dr. Lichter. Dr. Pfister directs the research group of molecular genetics of pediatric brain tumors. He is the head of the Division of Pediatric Neuro-Oncology, and he serves as the director of the Children's Cancer Center in Heidelberg. Dr. Pfister is one of the world's experts in pediatric neuro-oncology. His group was a pioneer at bridging genomic data, including genome, transcriptome, epigenome analysis, with integrative bioinformatics approaches and faithful in vitro and in vivo models for preclinical testing to find new therapies for children with brain tumors. A recent publication in Cancer Discoveries shows how Dr. Pfister and his group translated these studies into a pediatric oncology informed registry for patient care using genetic molecular signature of the individual tumor for patient selection and personalized cancer care, which we can certainly discuss with him at the end of the talk if you're interested. So for all this amazing body of work, Dr. Pfister published more than 400 manuscripts and received several prestigious awards, including the King Philip Award for Pediatric Oncology in 2009, the Alfred Müller Award for Neuro-Oncology in 2011, the German Cancer Award in 2012. And in 2020, he became member of the National Academy of Sciences, Leopoldina, and last year he received the Leopold Grifold Award. So without taking more of your time, Stefan, we're looking forward to your lecture, and the floor is yours. Welcome.
Thanks a lot, Martine, for the warm welcome, and I'm very happy to be with you, at least virtually today, for me tonight, for you in the early afternoon, I guess. And I'll just try to share my screen now to see whether this works. Again, it's a real pleasure to at least meet again in this format. I hope that we will also be able to have more personally meetings again anytime soon. I will just start with a little bit of an agenda to show what I will be talking about. On the one hand, this will be a very short introduction into childhood tumorigenesis, and I'm sure that this is very familiar to all of you, so this will be quick. In the second part, I will be talking about how we have been using especially methylation patterns or other epigenetic patterns for identifying cellular origin signatures and using this for tumor classification. And in the final part, I will try to give a little bit of an insight into a first rather superficial at this stage analysis of mapping single-cell tumor data onto single-cell normal human development, both in the cerebellum and in the brainstem, which basically will hopefully help us to further try and understand cellular origins of tumors, but also identify potentially more specific vulnerabilities of these tumors for specific therapeutic interventions. I think we can all agree that really the medical need is in these patients that still cannot be cured, and unfortunately this rate has more or less plateaued for many diseases over the past almost 20 years now. So I'm personally very convinced that we really need to define these very high-risk patients upfront for innovative first-line therapies rather than carrying on with non-effective therapies knowing that none of these patients can be rescued by some of the therapies we are currently applying. Also, just as a reminder, we are always facing this issue of really pediatric tumors all being very rare and additionally typically not occurring or rarely occurring in the same biology in adults, which basically always comes with the challenge that companies tend to not be very interested in developing specific therapies for pediatric cancers because the market is simply too small. And I'm just saying this because I guess all of what we are doing in terms of profiling tumors now makes yet rare entities even rarer, which can be criticized. But on the other hand, that's just a matter of fact that we have to accept that the biology is complicated and very diverse. And just as a final comment for the introduction, so one of the things that could potentially be regarded as a hallmark of pediatric cancer is really that very commonly these tumors seem to be caused initially by a maturation clock or hijacking of normal development as a very early step of tumorigenesis, which then arrests cells in a very primitive state and allows them to proliferate and keep growing and also invading other tissues, which is really, again, in stark contrast to most of the carcinomas in adults, which are thought to arise through successive accumulation of genetic hits over time derived from more mature cells to start with. So in essence, we are now facing a situation where we can distinguish almost 200 molecularly distinct brain tumor types in children and in adults. However, our standard diagnostic approaches do not nearly account for this heterogeneity, and even less so our therapeutic approaches. So I think at this stage, one could also conclude that, so why bother to split them into so many different groups? I think it's rather the other way around. Unless we really understand the heterogeneity within larger categories of tumors, we'll never be able to really help these patients who still die from their disease or relapse early on after treatment. So talking about molecular diagnostics, what we can do today is really to try and take an integrative approach. And this is something that also pretty much the 5th edition of the WHO classification of brain tumors that was issued just a few weeks ago has taken to not only think in different disciplines, but really try to collect information from the surgeon, from the pathologist, from the radiologist, but also importantly add a level of biological annotation to all of these tumor samples to really come to an integrated diagnosis. And this has traditionally been mostly done through microscopy and imaging. And now we have a whole repertoire of new methods available, not all of which can obviously currently be considered standard. But I think that tumor classification through methylation is getting more and more of a standard. And we urgently need to also have validated tools for clinical applications rather than research use only. But we can also systematically look into tumor drivers now through next generation sequencing approaches, into the host genome, into the microenvironment. And we can also start thinking of adding single cell and spatial omics more in a diagnostic setting rather than only for a research portfolio. And the same holds true for drug sensitivity testing, which really in many cases where we don't find a clear genetic driver or a clear target can probably help to identify rational treatments for patients for which we currently don't have a good idea how to deal with them. So why methylation? Obviously, we all know that every tissue in a human body in principle has the same underlying genome, while still the different cell types and different tissues develop. And this is largely dictated by the epigenome. And one readout of the epigenome, if you wish, is the methylation. And what we have seen really when looking into tumors that this is also true for tumors, that a large part of the DNA methylome is actually driven by a fingerprint of the cellular origin of tumors rather than something that undergoes massive evolution during tumorigenesis and during tumor progression. And that was actually for the first time realized in our hands, at least, when we had the opportunity to look at genome-wide DNA methylation patterns and correlating them with RNA expression at this stage, which basically turned out to be very interesting because in many cases we have seen that specifically the promoter regions of genes and their methylation status was really very strongly conserved between the suspected cellular origin and the tumor, both in human and in mice. These were also not regions where we saw a lot of differential expression between different tumors within one category, but rather in other regulatory regions. So we think that especially the promoter regions that we all at some stage at med school learned to be the places of hypermethylation happening in cancer, that much of this hypermethylation is actually a cellular origin fingerprint that does not change during tumorigenesis and cannot distinguish the cell of origin from the actual tumor, which is kind of explaining why many of these hypomethylated genes are actually cancer type specific, but not variable between different tumors within a biological category. At this stage, we were thinking of whether it would really be possible to come up with the largest possible reference cohort of all different brain tumors that you can think of, and then basically map an individual patient sample onto this large repertoire of different classes, of different brain tumor classes, and ideally give this mapping a score of confidence how likely it is that a certain tumor belongs to one of these groups that we have in the reference. And this is when we started out with the brain tumor classifier about 4 years ago by now, where we initially had around about 80 different entities annotated, and where we used a random forest artificial intelligence classifier system that basically also gave us a confidence score between 0 and 1 for a certain tumor, a specific single tumor to belong to one of these entities that we have reflected. And I think one of the strengths of this approach has always been that the classifier would also admit if it doesn't really know what it is. So then you would not get a good match, which is sometimes the problem with many of these tools that are around, and sometimes also with pathology diagnosis, that they are forced into a box without really knowing whether it belongs to this box. And we also, as many of you will know, have put this algorithm out to the web free of charge, which can be utilized on this website, molecularneuropathology.org. And by now we had about 85,000 uploads of methylation profiles, mostly brain tumors, but also sarcomas more recently. And in 65,000 of these cases, the patients and submitters basically agreed that we can make further use of the data for fine-tuning of the classifier, which basically means that by now we have around 100,000 profiles in the database, which we can really get down to very rare tumor entities that a pathologist would probably only see once or twice in their lifetime. So this is really something that basically is quite an amazing resource by now. From this world map, you can also see that people are using this tool now from all over the world, which basically also illustrates that this is something that can be actually thought as a tool that is capable of classifying tumors from all different regions in the world. And we have also gone on now, because that obviously is still all a research use only basis, to prospectively try and validate this, which we have done in a clinical registry study called MNP 2.0, where we try to enroll every newly diagnosed pediatric neuro-oncology patient, so a newly diagnosed brain tumor patient in Germany. Later on, we have also added patients from Switzerland and Australia and New Zealand. And really collect all the information, so the central radiology, the central pathology, as well as the molecular data, which basically was comprised of the DNA methylation analysis and a neuro-oncology NGS panel, both in tumor and blood. And whenever we found a discrepancy between the blinded neuropathology review and the biology, basically would trigger a molecular tumor board discussion about this individual case. And after enrolling more than 2,000 patients onto this, which has been population-based now, so 80% population-based for Germany for quite some time, Dominic has already started to analyze some of the results here. And one of the most striking results, which we have also seen in retrospective series, is that in quite a number of cases that were diagnosed as high-grade glioma in children, about 20% of what was called a glioblastoma, about 30% of what was called an anaplastic astrocytoma, molecularly look like low-grade gliomas. And if you now look at the survival data, and this is prospective and population-based now, you on the right-hand side see the difference between grade 3 and grade 4, as was done by reference pathology, which obviously stratifies the patients in the expected way. And on the left-hand side, you see how the molecular groups stratify the patients, where you, in a high-grade glioma cohort, have strata with 100% overall survival after several years. And the same holds true for progression-free survival. Whereas you also see these very bad-looking curves, such as the K27 mutant gliomas, which obviously are associated with very bad outcome. So that's really how it looks like in real life. So on the right-hand side here, this is at a meeting with Queen Maxima from the Netherlands. Basically, this was a patient from ours diagnosed about 8 years ago with a polycystic astrocytoma, retrospectively, but back then it was unambiguously called a glioblastoma by 3 pathologists. It was sent to a 4th pathologist who basically back then said, well, this could also still be a pilocytic, but I'm also not sure. So we treated her according to a high-grade glioma protocol with radiotherapy, temozolomide, and she obviously survived for many years, is studying business now. But really, these patients are, in question whether they would have needed all of this treatment and all of the toxicity that comes along with it. So this is really a question I think that the community now has to tackle and make sure that we are not overtreating a substantial proportion of these high-grade glioma patients. Just a very quick glimpse, moving away from methylation here for a second on the other data that we have collected on this, and just sticking with high-grade glioma because this has been the common topic here for the time being. One of the interesting findings that we have also seen in this prospective cohort is that around about 19% of the overall high-grade glioma cohort seem to have clearly pathogenic germline mutations in a number of different genes. And when accounting for molecular subgroups here, we also see that there's a very strong enrichment in some of them, including one group here that is characterized by MYC-N amplifications often. And similar to the MYC-N amplified medulloblastomas from the Sonny Ketchup subgroup, we also find quite a significant fraction of these patients actually having Li-Fraumeni syndrome underlying this diagnosis. So this just is one example in my mind to show that once accounting for these clearly different molecular subgroups, sometimes you also find associations that are really also clinically relevant. Just as a side note, obviously this is not specific, the methylation-based tumor classification is not specific to brain tumors. The principle also works in many other tumor entities. So this is an example here of the sarcoma classifier that was developed mostly at our neuropathology department. which is also now more and more being used for difficult to diagnose sarcomas. And there's also a classifier version on the website that can be used and describes all of the different classes. I just want to quickly also give some examples here of discoveries that can be made here by now looking into all of the very large number of tumors. And this is just kind of a fun example here, but also interesting because this is when we downloaded all of the TCGA methylation data as well and found two of the glioblastomas clustering with prostate, which basically very nicely shows that, again, a cell of origin signal can be very easily derived from them. And these two glioblastomas probably most likely were metastases from prostate cancer. So it's also extremely helpful for cancers of unknown primary if one is not sure where the tumor really came from. And then now focusing only on the brain tumors, we very often see these little islands that we don't know much about. And this is just one example here of one of these islands of 12 to 15 tumors clustering very closely together in unsupervised clustering methods. And in this case, for instance, these were 3 cases that were, or 4 cases that were annotated as gliofibroma in the beginning, which is an exceedingly rare diagnosis. So that caught our attention because this is so rare that we typically don't see that in any other group to be enriched. But at the same time, we also found that all of these cases clustering in this tight methylation cluster actually had a translocation between these 2 genes here, which we never had found in any other brain tumor, indicating that really this coincidence of a specific genetic hit and a specific methylation pattern that is very distinct from everything else is quite a recurrent topic. And this is really something that we have seen over and over again, that there seem to be specific lineages at a specific differentiation state that appear to be susceptible to very particular genetic hits, which also makes sense. We all know that if we try to knock out a certain gene in different tissues, some of them will not care and others will probably form a tumor. So at this stage, we also asked ourselves whether it was possible to also search the other way around. And this was actually the reason or the way how we first discovered the role of ALK fusions in pediatric hemispheric gliomas. And in the beginning, when we had this first patient here, which was 7 years ago by now, we thought that this might be a sample swap or something because we didn't expect any ALK fusion to really occur in a brain tumor. But then we used this case and said, well, if there's other cases that have a very similar methylation profile, would they probably also have a similar genetic hit? And through this, we identified, I guess back then, at least 10 more cases that had a very similar methylation phenotype and also were harboring ALK fusion. And then in a second step, we also found that other receptor tyrosine kinase fusions are also enriched in this group of patients. To this end, we have started to also collect more systematically clinical data on all of these patients, also including the patients that are basically analyzed through the website, because we think that this will be very helpful in interpreting the data even better than only, quote unquote, looking at biology. So this has started. And we also, in the WHO classification that was just released, we are very pleased that for all of the green entities here, methylation-based analysis is at least recommended for the difficult to classify cases, and for the orange examples, it's mentioned in the desirable diagnostic criteria. So quite a broad coverage now of this in the WHO classification. And what is also really nice about this new classification, in my mind, is that that for the first time, from a clinical perspective, it's also really helpful that a lot of recommendations involve potential predictive biomarkers that are not necessarily essential for diagnosis, but really are essential for clinical decision making. So this is really a big step forward that we have seen in this classification. And the other aspect that I just wanted to quickly mention here is that in many cases of newly added subtypes or tumor types, that primarily they are defined through their genetic aberration or sometimes through their methylation pattern, rather through a morphological pattern, which also illustrates the paradigm shift that happened here. Finally, just as an additional comment here, we also, for the first time, will have a WHO classification specifically for pediatric tumors, which I think is really timely to take them out of being hidden in all of the organ-specific books, but put all of this information together in one book. So this is currently being finalized and will be released sometime this year. Now, coming back to this question of hijacking of normal development and also some thoughts on how we can identify specific developmental targets, both for immuno-oncology and for maturating therapies. I just want to introduce a few data sets that we have generated and are currently working with. The first one being a single nucleus data set of around about 200,000 cells that we have generated together with Hendrik Heesman, who is an evolutionary biologist here in Heidelberg, where we tried to capture all the different time points of normal development, post-birth, and all the way into adulthood from 3 independent human donors and did single-cell analysis on them, and then came up with an atlas of normal human cerebellum development. We focused on the cerebellum initially because we thought that this was particularly relevant for pediatric brain tumors, and unfortunately not so well covered yet in the human cell atlas efforts. And what you can see on the right-hand side here is just a snapshot on how dramatically cell type composition is really changing between the embryonic phases and after birth in the cerebellum, where you can easily imagine that if you had identified shared targets between a tumor type and an embryonic cell state, that these might be really good targets for therapy as well, because many of these cell types will be very lowly abundant or even absent after birth in a mature body. You also try to complement this data with data sets, both RNA sequencing and attack sequencing from mouse and opossum, to also look into evolutionary trajectories and developmental trajectories against or between different species. And we have also collected a very large transcriptome data set from pediatric, mostly brain tumors, but also some non-brain tumors as controls to basically try and map these bulk transcriptomes onto the single cell types that we could identify in the normal cerebellum. And when doing so, and we basically used a combination of 2 methods here. One of them is just correlating expression patterns, and the other one is called gene set variance analysis, where you basically select the top differentially expressed genes in every cluster and then try to find hits. We basically could now correlate all of our bulk transcriptome data of all the different brain tumor entities on the x-axis against all the different cell types that we see in the cerebellum on the y-axis, and come up with some of the known hits, but also identify some new hits, or let's say correlations at this stage. So for instance, if you take the first column here, sonic hedgehog medulloblastoma, you get the best overlap with normal cerebellum in the granule cell precursor cells and also in more differentiated granule cells, which is exactly what we would expect. And for some of the other entities, it's equally clear, but not all of these were previously so clearly shown. So when zooming into medulloblastoma a little bit further, when now accounting for the different subtypes within sonic hedgehog and group 3, group 4 on the right-hand side, and again, doing comparisons between bulk transcriptomes and the single cell data from normal development, we again see in the first 4 columns that the sonic hedgehog medulloblastomas would mostly correlate with all shades of differentiation of the granule cell lineage, as has been established decades ago, and confirming data from Paul, Northcott, and others. We obviously also see that group 3-4 medulloblastomas mostly map to the UBC branch of this lineage. But there are some really interesting hints here when looking into single cell data, mapping these onto these normal cell trajectories. That on the one hand, we also get a lot of hits in different states of differentiation here for individual cells in a tumor along the GCP UBC differentiation path. And all of the gray cells here in panel C on the right-hand side are actually cells where we don't find a good correlation between the tumor cell and the transcriptome of any normal cell type. And there we think, I mean, on the one hand side, it's very possible that we don't have reflected all of the rare, especially the less differentiated stem cell-like cell types. But one thing that is really striking here is that when you actually subtract the mixed signal from these transcriptomes and then map them again, we get a much better mapping to the normal cells than when basically having this very dominating mixed signal in the equation. So this is something that we are more carefully looking at now. And also the other aspect, and this is also seems to be a recurrent theme now, that we very often see this gradient of differentiation within a tumor, but also within even different clusters of the single cell data within one tumor. So in terms of target identification, this is just an example here of trying to overlap sonic hedgehog medulloblastomas with granule cell precursors and come up with genes that are specifically expressed only in one tumor type and the matching normal cell type. And this is the gene, for instance, called CYYR1, which seems to be a very specific marker for GCPs, at least for the cell types that we see in the cerebellum, and seems to be a very specific marker for sonic hedgehog medulloblastomas across all of the different tumor types that we can look at. But when then looking into other normal tissues outside of the brain, we also see that it's quite highly expressed in many other tissues. So in this case, it would probably serve as a good lineage marker in the brain, but certainly not as a good therapeutic target because it would also be expressed in many other cell types. When taking a kind of a positive example in this regard, Claudin-6 here, which is one of the targets that BioNTech is currently generating CAR T cell programs against, we basically see how these targets look like. So you always have on the right of this dashed line and the adult tissue. So this is not expressed in any adult tissue, but it's really only expressed in a subset of tumors. So this is something that we would like to see here and ideally in even more tissues and even better resolution in terms of the development here for a good drug target. So we also have identified a few of these in different entities, at least candidates here. So one of them is called GPR17, which we found to be exclusively expressed in oligodendrocyte precursors or progenitors and pilocytic astrocytoma when overlapping these. And this is also specifically very highly expressed in these tumors. And not expressed, doesn't seem to be expressed, at least in the tissues that we have looked at so far outside of the brain, and is also very strongly downregulated after birth in the brain. And one additional interesting finding that we have also seen now in specifically in the pilocytic astrocytomas, and this is just one example here that I would like to share with you, where basically the red cells here in the left upper panel are the tumor cells that are projected onto the different normal cells of the target lineage, in this case, oligodendrocyte precursor lineage. We basically see that a large proportion of every pilocytic astrocytoma obviously is comprised of immune cells, which are colored in greenish here in the lower panel. And a larger proportion of the tumor is also quite nicely matching onto oligodendrocyte precursor cell differentiation. But what is even more interesting now is to look at individual pathways or genes here. And one of the interesting things that we found here is that many polycytic astrocytomas seem to have an appreciable level of expression of MHC class II on the tumor cells, which is quite specific, and we didn't see so much in many of the other brain tumors. What was really interesting was that MAP kinase targets in the tumor cell population are not really higher expressed than in the immune cells and in some of the other normal cell types. What we also saw quite significantly is that senescence markers such as CDKN1A are on the one hand expressed in tumor cells, which we have already known that many of these tumors undergo oncogene-induced senescence, but also at very high levels in the immune cells, mostly microglia that are surrounding the tumor, which we think is also something that will be interesting to be further understood. And to this end, this is just the medulloblastoma example, but we are currently trying to come up together with David Jones's group with a probe set for a panel of 100 genes for spatial transcriptomics analysis to really further understand this interplay. And together with John Maris, we are working on an immune peptidome analysis to try and understand what these MHC class II molecules on polycytic astrocytoma cells really present, and what the immune cells that also harbor MHC class II obviously present. And just as a final example in this category here, I also wanted to show some data on a mapping that we have done between tumors that were initially diagnosed as a medulloblastoma relapse, but turned out as a secondary glioblastoma radiation-induced, where we always, and I know that many of you have also had these discussions where we were always struggling with this kind of question of dedifferentiation or whether this is truly a new tumor. And we're now basically mapping these medulloblastomas, so primary medulloblastomas and secondary glioblastomas onto this normal cerebellum developmental data. We very clearly can distinguish the mapping between what is a medulloblastoma sample and what is a glioblastoma sample also in this regard. So it's not only that they don't share genetic aberrations, but they also really seem to come from a very different lineage, which is kind of expected, but it was a nice confirmation to see here. We also have developed an app which is online now to play around with this data, both for the bulk data, the transcriptomes mapping these onto the normal cell trajectory, but also single cell tumor data. So essentially, in this case, the red cells again are the tumor cells, and try to map them against the most closely matching normal lineage that we can identify, and to really also see these gradients of differentiation that we often observe in the tumor samples. And finally, and this is just an outlook because there will probably not be enough time to discuss this as well today, and we are only starting to analyze this data here. We have also generated a similarly sized data set for the brainstem by now, annotated the different cell types. And this is just a first example here of the DIPG single cell data set mapped onto this trajectory of oligodendrocyte differentiation, and again showing that there are cells and there's a bulk of DIPG cells that is mapping to a very early progenitor, but there are also cells that are much more similar to more differentiated cells within the same lineage, which seems to be really a common topic. With this, I'd like to summarize and hope that I could convince you about methylation fingerprints really showing a very nice tool for tumor classification through their reminiscence of the cellular origin. We see that this is more and more commonly being used also as a tool for the neuropathologists to aid their diagnostic decision-making. In several different studies, we have now seen anything between 10 and 15% of reclassification of tumors that would be clinically relevant in terms of really trying to these patients in principle needing a different therapy. We still are working on many more of these islands, and this is not only us, obviously, it's many different collaborations that we are interested in now to better understand. As I said, very often we see this close correlation between a specific genetic hit and a defined molecular group. In the space of pediatric high-grade gliomas, I think we have to address this question of whether these patients survive because or despite our therapy, and how we can basically ideally prevent them from overtreatment. I just want to again put out a plea that really molecular workup, and I'm sure that David Ellison and others would agree with that. Costing like 2% of a modern cancer therapy is really a worthwhile endeavor, and should not be a nice to have for sites in the world that can afford to do it. We think that these single cell and spatial technologies will further help to identify not only cellular origins of tumors, but also potential new targets. And we often see this gradient of differentiation within tumors, which was described in other ways before, but I think that this is something that also might be meaningful in terms of the tumors being able to form a tissue rather than a single cell type that is getting amplified. And again, this will probably also help revealing new targets in the future. With this, I'll stop. Thank everyone in my division at the kids, and also really want to especially emphasize David Jones's group here, because they have really contributed a lot to all of these studies that I have shown you, as well as Olaf Witt's clinical trial unit at the kids. Many collaborators from neuropathology and also everyone who has contributed to molecular neuropathology study, and a large network of people across the globe who have contributed to the reference cohort, and everyone who was working together with us in this matching exercise that I just showed you a few preliminary data on today. This I want to stop and just remind you again that we would love to see all of you in Hamburg in June, which hopefully will be one of the first meetings in our community to happen mostly in a live fashion again, which would be great. And with this, I hope that we still have time for questions.
Yes, thank you so much. It was wonderful. We can't see you, so I think we lost your picture, but I know you can fix it because you fixed it.
I will try to fix it again.
There you go. We see you.
There you go.
Perfect. All right. So there are several questions in the chat. So I'll start with the first one from Dr. Zhang. Expression is much more dynamic than methylation methylation. Could this cause any issue when matching single cell expression with bulk tumors? Especially the two are typically measured at different times.
I'm not sure whether I understand the question correctly, because we are matching gene expression with gene expression here, right?
So Dr. Zhang is, I don't know if Dr. Zhang can, answer. If he does, we'll get to him. Dr. Zhang, why don't you just, put make sure that your question, can be answered. So I'll go to David Ellison. A CHD7-VGLL3 fusion gene is present at high frequency in hybrid schwannoma, perineurioma, and rare intracranial examples occurs. Because the gliofibroma is so rare and its morphology is poorly characterized, could your gliofibromas actually be these hybrid tumors?
Yeah, that's very well possible. And this is one of these groups that we have not really put a lot of effort into further also annotating the morphology and the original diagnosis, because it's a rare group and they were collected from all over the place. But that's very well possible, and we'd love to work together here to basically probably increase the numbers to a reasonable size where we can also get a better clinical annotation of these samples.
Thank you. So there's one question from Dr. Zawistovsky. Any thoughts on molecular mechanism for the example you gave on the connection between your outlier island methylation clusters and the copy number alteration?
Yeah, I mean, I think that what is at least plausible to me, being this copy number alteration or being specific mutations or translocations, is that I think it's plausible that an evolutionary or selection advantage is also only conferred when a genetic hit happens in a certain cell type and not in another one, and this is why probably the same events will occur in different cell types as well, but they will probably not harm or will even harm the cell so much that it will undergo apoptosis rather than growing out as a tumor. Hope that makes sense.
Yeah, Stefan, we lost you again. You lost your pictures. We can hear you fine. So I'll go to the next question. Maybe your face. Here we go. You're back. So a question for Jana Rucci. Dear Stefan, thank you so much for your talk. I was wondering if you could tell me how you process the data when clustering different subtypes of medulloblastoma. I saw that Sonic Hedgehog, Wnt, and Growth3 for subtypes separated into different clusters. Did you integrate the patients from each subtype separately before merging all data together?
Yeah, basically the subtype annotations are derived from the. So initially we do a large UMAP analysis, but then when putting up the classifier, we basically try to identify CpG sites for each of these clusters that would best distinguish this particular cluster from all the other clusters. And this is what we use for annotation. So the best split really happens when we include all of the different tumor types that we can get a hand on.
Thank you. So the next question is from Dr. Jiao. Thanks for the great talk. With the technology development, the RNA-seq had been from bulk-seq to single-cell RNA-seq and spatial transcriptomic. Is there a possibility or need to have single-cell or spatial bisulfite sequencing for methylation study?
Yeah, I mean, we are currently trying to increase the turnaround of this and the number of cells that we can sequence. And I think that there's not a very good protocol out there yet, but we would be very curious to really look into the heterogeneity of methylation in cells, because we would assume that at least this cell of origin derived methylation patterns should be very similar between all of the cells in one tumor. But obviously this has yet to be proven. So we would be keenly interested in being able to generate such data. But currently this is more in the range of hundreds of cells rather than thousands or even 10,000s of cells where this is possible.
Thank you. All right, we have a question from Argentina. We see. So I'm not going to butcher her name, but hopefully not. But Edith Greenspanschel from Argentina, leading a foundation, kids with DIPG from Argentina are doing efforts to receive treatment in the Brzezinski Center, about which we have lots of bad references. What can you tell me about it?
Well, that's a difficult question. So as far as I know, this is a center that applies quite a variety of quite not scientifically validated treatments, and I think that's where the bad reputation comes from. But yeah, I have to say that I fortunately have not dealt with a lot of patients recently who basically went there. I think that's more for hype in America than in Europe at the moment.
Thank you. Susie Baker, hi Stefan. Previous single-cell studies indicated that DIPGs appear to be primarily comprised of OPC-like cells. Are you seeing a significant component of DIPG tumor cells with greater enrichment of neural stem cells and OPCs across many different tumors? So this is the first question. She has more questions.
Hi, Susie. Yeah, that's obviously a great question, and we, as you know, we have also proposed that really the cellular origin is rather much earlier than into a differentiated OPC lineage. So basically, with the mapping, we see all shades of differentiation into OPCs. We also see some cells that seem to map to very primitive stem-like cells, and at least my understanding would be that most likely the earliest node in this mapping that you can identify is probably the cellular origin, and maybe this differentiation that we see along a certain lineage within the tumor is something that could plausibly also happen after tumor onset. So at least this would be kind of a current working hypothesis, I guess.
So the next question, maybe it's related. Are some of the tumors that you analyze primarily OPC-like, as in the Philbin study? And is it possible that the profiles in tumors that you're analyzing are similar to the Philbin study, but the assignment of signatures to corresponding normal cell type is different?
Yes, I think we are pretty much finding the same thing. So I don't see a big discrepancy here. It's more the proportional composition that might be a bit different. But I think that the overall conclusion is the same. And I think the more striking or more relevant question now, and this is what Susie has just pointed out, is whether these transcriptome signatures that we see in the tumor, whether they are surely a reflection of the cellular origin, or might also be some acquired features of the tumor that basically helps them to survive and even starve in the brain environment. So this doesn't necessarily have to be a reflection of the cellular origin. And I think we have to be very critical since with this correlation, since currently it's not more than correlations. It all needs to be proven in the end.
So I see one more question from Rashid Drisi. How can you reconcile the possible sickle cell of origin of tumor with the intratumor heterogeneity seen with brain tumors?
Yeah, that's again a tough question. I think that one of the really interesting things to do that, and we are just starting this at the moment, will also be to really get multi-omic single cell data, and we have adopted a method now which basically we were hinted at by Bill Evans, where we can do whole exome and RNA sequencing from the same cells, which probably for the first time will really enable us to also try and understand how much the individual cell's genome, or at least exome, influences the transcriptome to help interpreting at least part of the transcriptome signatures that differ between different cells.
Thank you. One more. In your summary slide, you had mentioned 30 plus islands of novel brain tumor entities yet to be discovered. Could you elaborate on this? And how can one get involved in and possibly collaborate with you on this?
Yeah, so basically these are the islands. Some of them are actually already part of the version 12.5 classifier, which is now on the web. But we and David Ellison rightly pointed out that for many of them we are currently missing the best possible annotation and interpretation. So these are all still ongoing, and the way to collaborate there is basically just if people identify one of these tumors or even several of these tumors that map to one of these very rare groups that are reflected in the classification system now, to just get in touch and we'll make sure that we work together on these rare entities, because as said, it's much better to study 20 where you can have a chance to have at least 10 with a decent clinical annotation and remaining slides for pathologists to revisit them and stuff like that, rather than having 10, where then if you have a dropout rate of 50%, you end up with 5, where it will be very difficult to make any kind of firm conclusion.
So I have one more. Thanks for the mind-blowing talk. Does the correlation between molecular hits and cell of origin means that we can look at oncogenes as two groups? One group is the genes that are more susceptible for genetic alteration in specific lineage and therefore can be used for subclassification. And the second group is less specific and therefore probably less important in subclassification. Thanks a lot.
Yeah, I'm not sure whether this is a black and white distinguishing feature, but I think that that at least to me this makes a lot of sense, because there are certain genes when they are mutated, amplified, deleted, they would cause cancer in many different tissues. So they are very pleiotropic. Whereas there are definitely very specific genetic hits that only lead to tumor formation in one specific cell type, and then these obviously can also be more easily used in diagnostic terms.
Thanks. So we're almost at the top of the hour, but I have to ask you this question because I think it's really important. Maybe to make some comment of your new INFORM studies on personalized care that was just published recently.
Yeah, thanks for bringing that up. So one of the aspects there, at least for us, I know that we have quite a different medical system than in the US. Probably it will not be an issue at St. Jude because you are resource rich, but basically one of the consequences of now putting this art in the public domain is that we are very close to finalize a contract with our health insurances now for this to become part of the standard of care and fully reimbursed by health insurances, which I think is really a milestone in also translating this into care. And the other effort that we are currently trying to push is to get much better clinical annotation for all of these patients who undergo off-label or compassionate use treatment with a large real-world data registry, which tries to collect clinical data in a kind of regulatory grade or clinical trial grade manner, including monitoring and all the quality controls necessary, which will definitely help us to also identify interesting responses outside of clinical trials. And obviously, many of the companies who are interested in that are also interested, for instance, in collecting toxicities and side effects in patients who get off-label treatment or even prescribed treatment in cases where there is already a licensed drug available.
Yeah, thank you. I think it's wonderful. I hope this works out. So we are at the top of the hour. I would like to thank Dr. Pfister for his outstanding talk. We will close the official event, but we would like to invite all of you who are interested to stay on for a continued discussion for the Q&A. And before getting back to the questions, I would like to thank all of you for coming. I encourage you to visit our website and mark your calendar for upcoming talks. Next week, March 24, our own faculty member, Dr. Jiang Yu, will be presenting on targeting hidden drivers toward precision medicine.
Thanks very much, everyone, for attending and for the lively discussion.
So, Stefan, we can stay, but I think you nicely stopped your talk, and so we had lots of questions. So I personally don't have any more questions, but I don't see any other on the chat, so I think I'm going to let you go. There are things on the chat that you might want to check because I don't know if you can you see the chat?
I can, yeah, thanks.
Yeah, so please look at the chat because there may be some questions that, we were not able to go through. And then anybody can actually stay or contact Dr. Pfister directly, I suppose. All right, Stefan, I think we're going to let you go. Thank you so much. It was wonderful, as usual. And I'm looking forward to see you in June.
Great. Thanks a lot. It
Thank
was
you.
very fun.
Bye.
Thank you. Take care. Bye-bye.
You too. Bye.